everyone, my name is Naufira and today I'm going to be talking about a very interesting topic, lac-operon, which is like a revolutionary discovery of gene regulation in prokaryotes. Okay, don't get confused by the term yet. Before we head to the main topic, lac-operon, we'll understand what gene regulation is because most of you might be confused about what it is. So, yeah. So, you know, like our body produces antibodies to protect ourselves when an unknown virus or pathogen enters our body. But have you ever wondered, like, how the antibody is produced right when the pathogen or virus enters your body? Like, how is your body able to detect it? How is your body able to respond to the changes that happens to your body? Have you ever wondered that? Well, that is because of gene regulation, basically. Your antibody, you know, it is produced only when the virus enters your body. It is not produced when the virus is not there, right? This is a kind of protein that is generated in your body to protect your body only when it's necessary. This is gene regulation. Before we head to the really difficult part, let us understand what gene expression is. So, we all are different. I'm different, you're different, you have different skills, I have different skills, and we only show those skills when it is our time, right? Similarly, all genes are the same. If you have a DNA, genes are basically a stretch of your DNA, like a portion of it, like a small portion of it. That is your gene. So each gene in the DNA has different information that can be used for the synthesis of different proteins in different cells. Because like your stomach produces hydrochloric acid to digest food. Your eyes don't produce hydrochloric acid as well, right? Both of them has different functions. So different cells produce different proteins and enzymes by the information that is present in your gene. And this process of synthesis of protein from a specific gene by using the information in your gene is known as gene expression. It's simple. It only has two processes, transcription and translation. Okay. Transcription is nothing but the creation or synthesis of messenger RNA strand from your DNA. That is it. That is your transcription. This is done by an enzyme called RNA polymerase. Remember that. RNA polymerase is an enzyme that is responsible for creating an mRNA strand from your DNA. And from your mRNA strand, you synthesize protein. And this process is known as translation. Synthesis of protein is translation. And your gene expression is nothing but both these processes together. That is your gene expression. And the mechanism by which this process is applied in your body is your gene regulation. Gene regulation is the controlling of gene expression. Your body controls gene expression and that entire mechanism is your gene regulation. So then, what you must have heard lac-operon, but you don't know what exactly an operon is, right? So, an operon is nothing but it is the functional unit where gene regulation happens, where gene regulation takes place. It is like a cluster of genes that work together to give the information to your RNA polymerase enzyme. As I said, your RNA polymerase enzyme is responsible for the process of transcription. It collects the information from your gene. So your operon is nothing but a cluster of genes that work together to promote the process of transcription. That is your operon. And by creating a single messenger RNA strand, then your protein is synthesized with your RNA polymerase enzyme basically. So that is your operon. I hope you understand what an operon is. Okay. So this is the simple structure of an operon, okay? Your operon has like four main parts. That is your regulatory genes, your promoter, operator and structure genes. Okay, don't get confused yet. Your structural genes is where all the information is stored. Okay, let us consider the structural genes to be X, Y and Z. X has X information, Y has Y information and Z has Z information. which consist of different kind of information or enzymes that the RNA polymerase can use basically. Okay? Your RNA polymerase enters the promoter and enters the operon and it binds to the promoter. First it enters the operon and it binds to the promoter. Promoter starts with P, RNA polymerase starts with P, so you can remember that. So both of them are like close friends. So your promoter helps the RNA bind to it, RNA polymerase, and your regulatory genes help in the binding of RNA polymerase to the promoter. Understand? That is it, okay? And operator is basically just an on and off switch. It's an on and off switch. It's like a gate for your RNA polymerase. If your RNA polymerase is binded towards in the promoter, it's like a gate. If it opens, it can go ahead, proceed, collect information from your structure genes to create an mRNA strand. But if it's closed, it's not going to happen. It depends on the situation of your body. The changes in the situation, changes in the environment. When it is, oh my god, sorry. Sorry. Okay, when it is necessary for the operon to create that specific protein to fight that specific situation, the gate opens. So RNA polymerase can go ahead, collect information. But when it's not necessary, it remains off. The operon remains off. So from there, your mRNA strand is created and your protein X from X information, protein Y from Y information, and protein Z from Z information is created. Okay, I hope you understand what operon is because it's the basis of this and you need to understand what it is so you understand lac operon. So these are the components, like I said, your promoter, operator and structure genes. Now, E. coli, okay, that is your Escherichia. Escherichia, you pronounce it as Escherichia. So your Escherichia coli is basically a bacteria that is found in your intestine region, in your gut region basically. It helps in the digestion of food. Most of the time it's harmless, but there are some few E. coli bacteria that may be harmful. But we don't talk about that right now, okay? E. coli is a very unique bacteria. It eats whatever you eat, okay? So you eat carbohydrates every day and it actually absorbs energy from the glucose through your carbohydrate food. If you eat bread, it absorbs glucose from it. Usually glucose is the energy source of this E. coli. But sometimes when glucose levels are low and lactose is only available, like for example you eat breakfast but you don't eat any carbohydrate, like you don't eat bread, you just drink milk. So there's no glucose available, only lactose is available. So that is when your lac operon comes into the play. Lac operon is responsible for the metabolization of lactose. Instead of using glucose, this E. coli uses lactose. Why? Because lactose has both glucose and glycogen basically. So both of them are like, this lactose is broken down into two parts and glucose is used from lactose basically. So that is why lactose is what it uses when it's in a critical state basically. So your lac operon is a system that allows the E. coli to metabolize lactose and use glucose from the lactose as a primary source when lactose is not there. Okay? This is your lac operon. Okay? And it allows the bacteria to switch energy based on its availability. When glucose is available, it uses glucose. When lactose is available, glucose is low, it uses lactose basically. So, okay. That is why lac operon is actually a huge example of gene regulation. Gene regulation it's like a system that controls your gene expression only when the proteins are available to conserve energy. So your lac operon is a huge example for this gene regulation that completely changed the ideology of scientists for this gene regulation and to understand more about gene regulation. Your lactose and allolactose are two main things that your E. coli uses when glucose is not available. And this lactose and allolactose is what's responsible to turn your lac operon on. it usually turns off when it's not there and when glucose is available. But this is responsible for it to turn on, and that is why it is known as inducers. These are known as inducers. So you can see the structure of lactose. It consists of your glucose and your glycogen, which is, separated by the metabolization, and finally you get your glucose, which is used for energy. That is it. Allolactose and lactose is basically similar. It's not necessary to understand the difference because it's kind of complicated if you don't know chemistry. Lactose is basically, both of them is linked by 4 beta-1 galactosidase linkage bond basically. But over here it's in beta-1 6 linkage bond. Okay, I'm not sure if you all understand this, but it's not important in our topic right now. Okay. Structure of lacopron. Just like any other operon, like I showed you the structure, it consists of structure genes, promoter, operator, and regulatory genes. Your structural genes in lacopron is your lac-Z, lac-Y, and lac-A. Those are the structure genes. For now, just remember the names, okay? And your regulatory genes is lac-I. That is what you call it, the regulatory gene of lacopron. We'll see the structure. Okay, so let's understand the structural genes of lacopron first. Structural genes, as I said, is the part where it contains information for the RNA polymerase. So your lac-Z gene, basically it encodes for an enzyme called beta-galactosidase. Okay, so beta-galactosidase is like an enzyme that is present in your lac-Z gene. Okay, which helps in breaking down lactose and allolactose as glucose for usage of energy. Basically, that's its function, breaking down of lactose and allolactose. Your lac-Y gene consists of an permease membrane protein. What it does, okay, by the name itself you can understand, permease. It sticks to the cell membrane wall, and allows the transportation of lactose or allolactose inside the cell. It's like it identifies lactose and allolactose when glucose is deficient and helps it to enter the cell. That is its function basically. And lac-A gene, basically it encodes for an enzyme called transacetylase. Basically, the function of this specific gene is not clear yet. It is known that it releases an acetyl group, but the function is not clear, though people and scientists has assumed that this is responsible for blocking all the unknown necessary compounds or substances that enter to interrupt this process happening in your lacopron. It basically protects the lacopron's process and protects it basically, but the function is not clear basically of your lac-A gene. Okay. Okay, fine. Slide is not loading.
You want to play?
No, it's fine. No, it's just one slide was not loading, that's why. It's okay. So this is your lacopron. So I've prepared a model for your lacopron. Basically like any other operon, okay? This here is your regulatory gene. These three are your structural genes, lac-Z, lac-Y, lac-A. This over here, this yellow part you can see, is your operator, the on and off switch basically. And this here, the white part you see, is your promoter. This is where your, this is your RNA permease enzyme. This is where it binds. This is where it remains in your lacopron, okay? So basically what happens, when your RNA polymerase is in your lacopron, and when lactose is available, lactose or allolactose, you have a repressor. Repressor is basically binded to your operator like this, okay? It is binded to your operator, so your RNA polymerase won't be able to pass further. Your repressor is a kind of protein that is released by your regulatory gene, LAC-I. Okay? LAC-I means inhibitor. Inhibitor means stopping the process. That's what inhibiting means, like stopping it. So basically, LAC-I releases this repressor protein, which always binds to the operator like a switch, which blocks your RNA polymerase from going further. That is the purpose of this repressor. Basically, when it's in its active state, it blocks. It likes to block. It likes to block the RNA polymerase and it hates the RNA polymerase basically. So, when lactose is present, your repressor loves lactose or allolactose. So, when lactose is present, it changes its shape and it binds to the lactose and it's taken away basically, which opens up this operator region so the RNA polymerase can pass through it and reaches the structural genes, which is your LAC-Z, LAC-Y and LAC-A. You all know what the function of these structural genes are. So RNA polymerase, grabs the information from these structural genes and creates your mRNA strand through which your protein is synthesized. This is said to be the on state of your LAC operon. When the gate is open, when the repressor is not there, has already been taken by your lactose and allolactose, it is said to be on. So your protein can be synthesized. But it is said to be off when your RNA polymerase is blocked, basically. And it's mostly always in off state. It's never usually on because glucose is mostly available. It only turns on when glucose is deficient and when lactose and allolactose is present. Okay? So that is your LAC operon. I hope you understand LAC operon. Okay. So this is all about LAC operon. So basically, for your better understanding, this is your repressor. Your repressor hates your RNA polymerase and your RNA polymerase is struggling to go further. It's binded to the promoter. Your operator is where your repressor binds to, blocking the RNA polymerase so it won't be able to go. Then when lactose is available, lactose changes the shape of your repressor, takes it away. So it's finally open and your RNA polymerase can go further, create your mRNA strand. This is it. This is a very simple way of understanding this LAC operon basically. Yeah, this is like the structure given in your textbook. It's quite confusing when you look at it from your textbook point of view. So now you can understand it basically. These are the structural genes, Z, Y, A. Your operator where the repressor is blocking, promoter where your RNA polymerase binds to. This is your LAC-I, regulatory gene that releases the repressor. Got it? Okay. This is it. This is your off state and this is your on state when your mRNA strand is created and protein is synthesized. Okay? So this is how, for your better understanding. This is the DNA. This part, a portion of it, stretch of it, is genes. Different kind of genes and different parts of your operon, LAC operon. Negative regulation. You understand what gene regulation is? Negative regulation is nothing, like I explained. It is when the regulation process is off. It is when it blocks the process of gene expression. It's when it stops it. It's when it inhibits the process of gene expression. So protein is not synthesized because it doesn't need protein at that time. That is your negative regulation. It's negative when it's off, basically. That's it. So your function of LAC operon, when it's off without lactose, when glucose is present, or when lactose is present but glucose is low, and operates to produce enzymes for lactose, so it can be used for energy. Lactose is used as a source of energy in your LAC operon, and it helps the E. coli to gain energy from lactose when glucose is not there, basically. This is it. Your on and off state, like I said, no lactose, off state, lactose, on state. So your lactose and allolactose is called inducers, like I said, because it is responsible for this lac operon to turn on, basically. So your lac operon helps you for understanding gene regulation very well. It is a very easy way of understanding gene regulation and very unique way actually. And its discovery changed everything for the scientists to understand more. And it helped in genetic engineering as well. It really helped the scientists to understand this concept. See, like applications in biotechnology, it helps in creating gene switches, which means that scientists use this as a source of information. So they can create an on and off switch, and they can create something new with that idea into other operons and other cells. They can use it to, use it in other cells as well, basically. And it helps you in genetic engineering and research purposes, basically, like I said. So that is it about your lac operon. So very interesting topic, right? Okay, thank you. I'm sorry if I took too much time.
Any questions? You don't ask too many questions. Where if you really could not understand any point is there.
Yeah, any questions?
Ask
What
first.
is lac A?
Lac A. Lac A is basically an acetylase. It encodes for an enzyme called acetylase. What it does is that it releases an acetyl group to control this gene regulation process, protect the lac operon from other outside sources that blocks this process. But still it's not clear yet about this process of lac A because scientists still have not figured it out. It's an assumption. Okay, anyone else? Yes. Yeah.
What happened? Can a mutation take place in this lac operon mutations?
Like a mutation.
Yeah.
Okay, yeah. Sometimes mutations can happen in lac operon, and that is why some E. coli bacteria are harmful. Yeah, because some E. coli bacteria, when you take in too much lactose, take in very less glucose, too much lactose but no glucose at all, like glucose is not even available, there will be some problems with your lac operon. It will be too excess for the lac operon to function properly, making the E. coli more harmful and can cause a lot of diseases through it. Basically.
What are the key differences between situation when the lac operon gets turned on and turned off?
When it gets turned on, okay, and turned off, basically. Key differences between this. When it gets turned on, it is called positive regulation, okay. This is when lactose and allolactose is available for energy, glucose is low, it is deficient. So this is used as a source of energy. But when it's turned off, glucose is already available. Your E. coli does not need to use more energy to break down lactose and allolactose. So using glucose directly for its purpose and turning it off.
Could you tell me the difference between two types of operons?
Two types of operons. Okay. There aren't many operons found in a lot of places. Basically, it is present in most bacteria found in prokaryotic cells. This one is mainly in the human body, but there are many other operons that are present in other organisms and other cells basically. And those lac operons, as I said, all lac operons is the same structure only. Same divisions, promoter, operator, structure genes. But your structural genes is different for every operon. Your every operon is different clusters of genes. Different genes, as I said, consist of different information. Unique information. For that, only for that function of that specific cell, because that cell is different from other cells. Yeah.
Are there any names for different operons?
Yeah, there are names, but I'm not familiar with a lot of operon examples, basically. This is, yeah.
What about repressor proteins? Do they have a specific name?
Repressor proteins is basically known for liking to block. It's a protein that is synthesized by a regulatory gene called LACI. It's a protein that is specifically generated when glucose is available continuously. So this protein is continuously generated, it's non-stop, continuously generated by a LAC-I regulatory gene. So it turns the lac operon off effectively and helps in this process to become effective, basically. Okay? Thank you. Okay.